So this part of the talk is about the kind of research we do in the lab and how we actually, it's a tale of two cells essentially. One cell that is resistant to a chemotherapeutic drug and another cell that is sensitive to a chemotherapeutic drug. The model system we work on in the lab is glioblastoma. So it'll be around temozolomide resistance. And temozolomide is the only available chemotherapeutic agent. It's called Temodar that is available for treatment of glioblastoma. So I will start my presentation, and before I continue, I would like to thank Shaon and Mohit and all the other students who've been behind the scenes to set up this wonderful, workshop. I've not been there for the whole length of it. I just came in last night, but everything, all the organization has been perfect. So thanks a lot to, both of you and to ICTS for hosting it. So as I begin, I'd like to draw your attention to this Pulitzer Prize winning book. It's a whodunit on cancer by the eminent clinician and scientist you probably all know of, Siddhartha Mukherjee. So one of the quotes in this book is, how can one capture genes that behave like ghosts, influencing cells from behind some dark curtain? So in my opinion, the author is referring to the fact that although cancer is indeed well known to be a genetic disease, there are other downstream factors, like there are other molecular hierarchies of the cell and their crosstalk that defines the emergent pathogenesis and outcomes of the disease. His other quote in the book emphasizes how cancer exploits the fundamental logic of evolution and the fact that we are a product of Darwinian selection, and so is this disease that lurks inside us, makes it really scary. So this gives a right of way for the methods that I described earlier for constraint-based modelers to draw upon the optimality principles of flux balance analysis to understand the emergent properties of cancer. So this is going to be the outline of my talk. It's a combination of thoughts that are described in that book. And they will essentially talk about combining technology, computation, and in order to eventually predict biological function. So I will showcase how using technology, including exome sequencing, transcript abundance, metabolite profiling, respiration growth profiling, etc., combined with computational analysis of the kind I showed earlier, some data-driven analysis, can come together to understand potential mechanisms of drug sensitivity, and can we predict chemosensitivity and come towards a more integrative paradigm, an integrated paradigm of resistance, and essentially understand the systems biology of glioblastoma. So cancer, as we all know, grows when a single cell begins to grow out of control. There are many hallmarks of cancer, through more than a century of research. These include the ones mentioned here. This is from a review that is very famous by Helen. So they include limitless proliferative potential, insensitivity to growth signals, angiogenesis, and so on and so forth. Surprisingly, although Warburg, way back in 1923, established that cancer cells produce a lot of lactate even in the absence of oxygen, it's been only quite recently that metabolism has become an emerging hallmark. And in my opinion, this is probably because only now researchers have begun to establish links between metabolic alterations and the non-metabolic hallmarks of cancer and the vice versa. So these arrows that you see in this figure, right, the centripetal arrows that go outward, they essentially tell you how the hallmarks of cancer impinge on metabolism, and the centripetal arrows, that is the arrows pointing outwards, the centrifugal arrows, the ones that are pointing inwards, illustrate how tumor-associated metabolic programming can contribute to the acquisition of these hallmarks. Now, as depicted here, cancer is a complex multifactorial multi-hit disease, and it is complicated by metastasis and recurrence. And by multifactorial, I mean that there are many factors that impact it, including the environment, the pH, the oxygen, the chemotherapeutic drugs that are being used. And by multi-hit, I mean essentially, if you look at the top over here, right, it brings about changes in the physiology through changes in DNA, RNA, protein, and also the metabolite levels. And so in order to understand the emergent properties here, one needs a very systems or holistic approach. Now talking a little bit about our model system, glioblastoma. So cancer can essentially affect any type of cell in the body. Glioblastoma is one of the most common brain tumors that starts from brain cells and demonstrates infiltrative growth. So essentially this means that it's like mixing salt and pepper together. It's very hard for you to separate them out. So essentially the incidence is very high. In people who are supposed to be in the prime of life, that is 45 to 55 years old, and the median survival is only 15 months with the current therapy. Some of the hallmarks of this of glioblastoma include increased metabolic demand, necrosis, and microvascular proliferation, and therefore, new strategies for treatment are imminent. As I said, the prognosis is really is poor. Treatment includes surgery, radiation, chemotherapy. The drug that I mentioned earlier, temozolomide, is the trade name is Temodar. There's a lot of tumor recurrence that occurs in glioblastoma, and therefore it results in further surgery. And it's an iterative process, very sad one. Very few experimental therapies have come out. Just recently, last week, there's been a new CAR T cell therapy that was, developed and came out through University of Pennsylvania. And generally, the mean survival is only 15.4 months. Therefore, new treatment options are desperately needed. Now, let's talk a little bit about temozolomide. So, temozolomide is a DNA methylating drug. Temozolomide is a prodrug that spontaneously converts into the active form, which is MTIC. So MTIC provides methyl diazonium ions and therefore creates methyl diazonium ions, and it's the methyl groups that get transferred to the N7 and the N3 position of guanine in the DNA, and that's how this drug works. But the sad story is, although there was a significant improvement in the survival of patients, if there is a lesion in this gene, the MGMT gene, the methyl guanine DNA methyltransferase, then you can literally reverse the process, reverse the effect of temozolomide, and therefore, people again get resistant and the survival goes down. So generally, as seen in this Kaplan-Meier curve here, when temozolomide is used with radiation therapy, it increases the mean survival considerably. But within a year of its use, people began to see resistance effects. Now, coming to the system that we use in the lab, we have a very interesting story. So we started off because of a project that was initiated before I joined my institute. It was an Indo-Japanese collaboration. And so when I came into NCL, I was told, hey, Anu, why don't you work on glioblastoma, and why don't you look at this particular cell line, U87? And since a lot of my training and my initial love was about metabolism, I decided to first tackle that aspect, come to that most outermost layer of the model, right? I could make that outermost layer of the model, the matrix, and I could also go in the lab and I could estimate all the metabolites that I could measure both intracellularly and extracellularly, and also probe what is the rate of uptake and rate of change of these metabolites inside the glioblastoma cell. So I told one of my talented students to grow this U87 cell line, and then every week, and these cells grow very slowly because they are neuronal cells. They are very delicate, and they have a long doubling time, so they grow very slowly. And every week she would come back to me and tell me that the cells are dead, the cells are dead, the cells are dead. And my prior training was in looking at infectious disease, looking at E. coli, and so on and so forth. So I didn't at that point quite understand mammalian cell biology or mammalian cell culture technique, and I kept asking her, why do you say this, Ria? I think the cells should be growing. How can they die all the time? You're an experienced cell biologist. And then she told me that the cells were floating on top every time she tried to grow them, because for doing metabolomics, like I said, there are always constraints, right? Nothing is really easy. It might seem easy once the paper is done, but it's really not that easy. So to do metabolomics, to do fluxomics, you need to get a large number of cells. So you cannot just do it with growing a small flask. So you need to get, you need large quantities of cells, because the metabolites are present in picomolar or femtomolar levels. So, and you want to estimate them. So while she was growing these, their cells would float on top. And I said that it cannot be that the cells are dead all the time. Let's do a dead or alive cell assay. And so she did that, and lo and behold, the cells were alive, they were not dead. Then the next question we asked was, hey, if these cells float to the top, maybe, we were quite naive at that time, right? I was just starting off as a PI on cancer. And so the question we asked is, are these the cells that have the potential to migrate? Are these cells, do these cells have drug resistance properties? Do these cells have stem cell-like properties as well? Can they redifferentiate into stems, into the neuronal cells? Because the cells that floated on top, surprisingly, had a different morphology when we looked at them under the microscope. They looked more circular, and they were more like spherical blobs as compared to the traditional neuronal star-shaped morphology of U87 or a neuronal cell. And then we decided that, hey, indeed these cells, so we went on, we saw that those cells were alive. Then we went on to the FACS, sorted these cells, got these two populations out, and then, as any typical biochemical engineer would like to do, we decided to do some growth curve experiments. And we decided to calculate growth rates and doubling times, because, hey, we wanted to quantitate this difference, right? And then we saw that in the mixed population, the doubling time was about 36 hours, whereas for the separated U87, which will be always shown in blue, the growth rate was, the doubling time was close by, it was 35 hours. But for those spherical cells that kept bugging us, we saw that the growth doubling times were quite higher. They were about 47 hours. Then we decided to check how these cells perform in the presence of different concentrations of temozolomide, and identified that there was a 40% difference between the IC50 values of U87 and we call these spheres neurospheres for lack of imagination, because they come from a neuronal origin, and they are spherical in shape, so we call them neurospheres. And my student decided that all curves plotted with neurospheres will be red, because they seem to they are more resistant to the drug, and hence are more dangerous. So that was her philosophy of the color choice here. So everything that is in red is going to be neurospheres. The cells that are resistant to the drug, to temozolomide, and everything in blue is going to be U87, the cells that are sensitive to temozolomide. So now that we established their drug resistance profiles, we decided to look at, hey, can these cells redifferentiate back into the neuronal morphology? And after adding the differentiation factors, within 4 days, we saw that yes, these cells were able to redifferentiate back. And then we went on later on to look at some cell surface markers and do some antibody tests to identify that indeed these cells were cancer stem cells.
So, when you did this ICP, how long you have done?
So we did, so the first set of experiments were done for about, I think 24 to 48 hours, then we increased it to 72 hours. So we did a kinetic profile as well as a static profile, because we realized that there could be differences based on the time of incubation of them. Yeah, so this is for the 48 hours, I think. So then once we got this understanding of the phenotypic difference, right? This was our primary phenotype, a differential drug response and differential growth. We wanted to connect it to what was the DNA sequence variation. So look at the genotype, and also understand, are there differences in the secondary phenotype as well. For us, we define some of the secondary phenotypes as respiration, because in mammalian cells, respiration and growth can be decoupled. Metabolism, because of course, I love metabolism. Nutrient preferences, because you can't have metabolism without having an array of nutrients. And nutrients also define microenvironments, right? So if the cell has to, we all know about or have heard about tumor heterogeneity, and if different, so glioblastoma apparently has 17 different cell types in the brain tumor. So if you were to take a tissue sample from a patient with glioblastoma and look at the individual cell types in that tumor, you would find 17 different cell types. So, we definitely felt that there could be changes in the microenvironment related to metabolites and nutrients that could change the way these cells behave.
Cell line that you started with is that 287, right? And then during the course of the reaction, like when she was culturing, she found out these neurospheres.
Yes. So so when she was culturing it, we found that the cells always floated to the top, and then we did an assay to understand whether they were different.
They were told when the cell line was born.
No, so then we went and did some more research on all, because these are 0.001% of the total cell population. So if you grow a flask of U87, 0.001% of the cells will be these neuronal spheres. And if you keep culturing them like we did because we wanted more cells, then more and more numbers come up. So we went and looked at, and these are called site populations. These site populations exist in all cancer cells. We went and checked A549, which is a model cancer cell line, lung cancer cell line, MCF7, which is a model breast cancer cell line, and so on and so forth. We checked 5 different cell lines, and all of them have a floating population if you grow them up longer. We did not sort all of them for lack of funds. But yes, they definitely, so a lot of cancer cell lines that we purchase from ATCC or from the European Association, they would definitely have some site population, very low numbers, but these could be the reason that cell line experiments don't really match the real patient information. Using FACS. So we use cell sorting, we add dyes, we use cell sorting, and then we reculture them separately.
You can see that from neurospheres, I can also get the cells.
Yeah, so you can differentiate them. So I showed that U87 had 756 micromolar as its IC50. The neurospheres, the spheres that we separated out using FACS had 1000 plus as the IC50. If you redifferentiate these neurospheres into neuronal cells again and make them adherent, then their IC50 decreases. It's somewhere in between what U87 and NSP has. So it is very complex, and yes. So then, we were quite naive, as I said, and so we were like, hey, maybe if we look at the genotype, if we sequence the genome or the exome, we will be able to understand the origin of these cells, right? Did these neurospheres arise from U87, or were they already there as a population? There is a neuronal stem cell, and did both of these arise from the neuronal stem cell, right? And we thought, hey, by doing this genotyping, we should be able to miss the butt. But I'll tell you later on that we were not able to. And then the other things we were interested in are these secondary phenotypes. And we also looked at chemosensitivity as also, what are the different chemosensitive agents and how do these two cells differ? That was also a secondary phenotype that we were interested in. So then starting off with metabolites, we did metabolomics and we looked at both exometabolome and intracellular profiles. And this is a discussion of the exometabolic profile, and I will just show you here two important or one important result. In the case of U87, we found that 4 compounds were rate limiting, were growth limiting, sorry, and for the neurospheres, only 3 of them were growth limiting. So we see that for U87 requires glucose, glutamine, serine, and tryptophan, whereas the neurospheres requires only glutamine, glucose, and serine. So tryptophan was one compound that the neurospheres did not need. They actually never used them. If you look at this heat map, they didn't use them till about day 8 or so. But after day 8, we see they begin to get consumed, and then they are consumed later on. They'd never go to 0, so they are not growth limiting, although and we've synchronized these cells for cell cycle to make sure that there are no differences in cell cycle that sort of induce these differences in metabolic consumption. The other thing, as I told you, we were interested in is nutrient preferences. So there are these micro profiling arrays that are available, which are called biolog plates. And these biolog plates essentially are in a 96-well format, and each well has a different carbon or nitrogen source or a different ion, and you can probe how your cell utilizes these particular compounds in isolation. So it's a proprietary, it's a proprietary composition. They don't tell you much. They don't even tell you concentrations till you go and call the CEO and ask them to tell you what concentration because you have 3 different concentrations at which you check the growth of the respiration of these cells, because it might be too high or too low to have an impact. So this figure here is a representation of growth of U87 and NSP on 92 carbon and nitrogen sources. And as you can see here, again the blue represents U87 and the red NSP. There are differences in the way glucose is utilized, the way citrate is utilized, hexanoic acid is utilized. And so we looked at each and every graph that we generated through this process, and we identified that there was a huge difference between the substrates, the number of substrates that were utilized for growth and respiration between U87 and NSP. So NSP can grow and respire on many more substrates than can U87. That's the take home from this slide. So U87 can respire on many substrates, NSP can grow on many more substrates. Now, what's the difference between respiration and growth? Primer is that when you're measuring respiration, you can look at all the cellular reductase activity that defines your respiration or your respiratory coefficient. Growth, on the other hand, is the doubling. So in those plates that we were measuring the respiration, the cellular reductase activity, we also went on to check how many doublings that happen by certain counting methods and tests that we did. And essentially our conclusion was that the neurospheres couple respiration and growth, whereas U87 does not. So then we went on to look at whole exome comparisons using the genes that are mutated in both U87 and NSP. And what is seen is that the 2 cell populations are riddled with lots of mutations, as you can tell here in the slide. However, there seem to be a sizable that are unique. And that do not belong to either the DBSNP or the cosmic database. And in this, since our interest was in metabolism, we saw that 7% of the total mutations observed belong to genes of the metabolic class. So this figure here also gives you, gives you an overview of the different genes mutated in different chromosomes. And you can see that there are there are quite a bit of differences. There's a lot of commonality and there are a lot of differences. So here in this talk, I will focus on two classes of pathways where I'll show mutations. Yeah?
If there is this much of chromosomal difference in mutations, my feeling, how come these cells can come from each other in some cycles of growth? Like if I'm growing them in dish.
No, they have not come from the dish. They are not artifacts. They are already there. They are.
When I facts isolate them,
yeah.
Again I grow them.
See,
how can I see them again immediately?
See what immediately?
Do I see both the populations again?
No, we do not see. If you grow them a lot, you probably will, but we make sure that so these passage numbers are very important. So initially we get the cell at say passage 10 or something from ATCC. So we make sure that we are not repeatedly passaging them and using the further passages for the experiments, because that will create more confusion and more heterogeneity. So we basically just grow them once or twice, and then we freeze down hundreds of tubes, and then we are always working with that initial passage number or the initial population. So it is not an artifact of laboratory use that we create these cells in them. So they are there, and probably because the patient, this is from I think this particular cell line is from a 64-year-old woman who died of brain cancer. So she was probably treated with some drug. Temozolomide was not there at that point, I think. So she might have, created heterogeneity, and this might have always been there. But so this figure here shows that there are 11,000 mutations that are common between them. So, but again, there are 559 that are unique for neurospheres and 397 that are unique for U87. So it's hardly conclusive that where the origin could be. Yeah. And between the metabolic genes that we were interested in, there was 15 that were unique in U87, 44 unique in NSP. So NSP seemed to have more unique metabolic mutations than U87, so which was exciting for us. Yeah, so this figure essentially shows you the mutational profile of growth factor and signaling related genes that are involved specifically in metabolism, and it might not be comprehensive, okay? So for those of you, how many of you are familiar with mammalian cell growth? Okay, only a few of you, again the 5%, right? So for those of you who are new to mammalian cell metabolism, mammalian cells are not cell autonomous for metabolic uptake and growth. So for example, if I have an E. coli cell in one dish here, and if I have a mammalian cell in another dish here, and they are both, say, in a state that they are very hungry and they're going to die, and I put glucose feed glucose to both of them. My E. coli cell will start consuming it immediately, but my mammalian cell will still wait because it hasn't received a signal. So mammalian cells are not autonomous for picking up metabolites. They need these receptor tyrosine kinases to be turned on, and then to start to give them the signal that they have to uptake this glucose, and then they have to start eating. For all metabolism to take place, for all growth to take place in a mammalian cell, these receptor tyrosine kinases, these signaling molecules are very critical. So a few take-home messages from the slide here are that glucose metabolism, which is generally controlled by that PI3K AKT pathway, is mutated. The negative controller, which is P10, is mutated only in NSP. It is not mutated in, yeah, P10 is here. Yeah, so again here the blue crosses show mutation in U87, red crosses show mutation in NSP. If you have larger crosses, that means there are more number of mutations in that gene in that particular cell line. Although here everything is looking similar except for S6K. So yeah, I'm not sure that statement holds here, but yeah, generally the way we made these figures were anything that has a larger cross has more mutations than the other. So here this is mutated, so this is for glucose metabolism. If you look at amino acid metabolism, which is also critical for mammalian cell growth, TSC1, which is the negative controller of mTOR, that is mutated only in U87 and not in NSP. Otherwise, you see mutations distributed across U87 and NSP. And eventually all of this, right, would tie up with regulation of oxidative phosphorylation, because the P53 is mutated in both, but then again P10, which controls P53, is mutated only in the neurospheres. Now another pathway that I find very interesting and shows a lot of mutations is the electron transport chain, which is connected to oxidative phosphorylation. So this one too is absolutely full of mutations. Now, bringing you back to high school biology or maybe 12th standard biology, the electron transport chain can be considered to have two different functions. One of it is the transfer of electrons, the redox transfer of electrons from NADH, right, the first complex here, to oxygen, to molecular oxygen. And the second is the conversion of the free energy of that electron motive force into a proton gradient, which eventually gives you ATP, right? So there are two functions here, NADH transferring electrons, electrons being transferred from NADH to molecular oxygen, and then making of ATP because of the changed electron motive force and the proton gradient. Now, if you look at the mutations, the mutational profiles, the pink ones represent similar mutational profiles. So every complex in the electron transport chain is coded by multiple genes, and you can see that it's riddled with mutations throughout, right? But what is interesting is that the ATP synthase seems to have a mutation only in U87. And the ATP4A, which is ATP lyase gene, is the ATPase gene, has a mutation only in NSP. So essentially, you could so what we did was we hypothesized that it is these mutations that sort of impact that differential growth and respiratory profile that we saw on those microplates, where we had those 367 carbon and nitrogen sources. No, I said that the TSC1 is a negative controller of mTOR pathway, and that is the one that's mutated. No, so the only thing I commented about S6K is the number of mutations in NSP are much higher because you have a higher cross here. All this data is deposited in the NCBI database for anyone to go and look at. Sure. ATP consumption. We haven't checked ATP consumption, but we've looked at some assays to measure ATP levels, and they are different in both these cells. NADH and NAD levels are different in both these cell types. These are also dependent on the carbon and nitrogen sources that you use, so they are substrate dependent as well. It's not a one-to-one correspondence, unfortunately, because as you see, there are so many mutations over here. So in complex 1, the only unique mutation is the NDUFS7 in NSP. There is no unique mutation in U87. We see a differential response of NSP two drugs that impact complex 1 like rotenone. Similarly, if you look at this complex 2, SDHA is mutated only in U87 and not NSP. So you have a differential response here as well to electron transport chain inhibitors of complex 2. So if you look at each complex, right, there is something that's unique for one cell or the other. Now, how do you look? Yeah. Yeah, I mean, even Psych1 is famous. I mean, everyone is famous. Yeah. So so it's very hard to bring in a one-on-one relation. You can only, especially you can't really delineate causation only from this mutational profile. Maybe correlation, yes, but causation, no. Right, then we decided to see if you have different doses of the drug, how does the cell grow in the presence of different doses of the drug. So we had one profile where we looked at no drug, that is what I talked about so far. Then we looked at a very small amount of drug, 10 micromolar temozolomide, 100 micromolar somewhere in between, and a 700 micromolar. As you can see, higher the concentration, the cells die. What was interesting for us was the 10 micromolar. So you can see that there's an increase over here. There's no death in the populations. And what we saw was that if you look at NSP population, you could see cells at this low concentration of temozolomide actually differentiate. So this seems to be, we haven't probed this further, but this is, I think, a cause of worry for a lot of clinicians, and I've talked to a few of them, because if you have dormant cells, these NSP, as I showed you here, has a doubling time of about 45 hours, which is much higher than U87. If you have heterogeneity in a tumor cell, in a tumor inside your brain, and these guys grow really slowly, the more dormant ones, the stem cell characterized ones, they can actually differentiate and create more tumor mass as time goes by if you are treated with suboptimal concentrations of the drug, right? So, and this has been seen as well to happen in bone sarcoma as well. So, yeah. So there's a lot. I think I was talking to somebody during the break. There's a lot of heterogeneity. There's a lot of there's a lot of reprogramming that takes place at different levels, and therefore it's very hard to answer your question as well. That's why when I started, I made sure to have a slide that says that it is a multi-hit and a multifactorial disease. Many factors that impact it, and there are many both molecular and physiological changes that happen during. So then, we looked at what was the differential metabolic profile in the presence of temozolomide across those different concentrations that we were interested in. And the take-home message from this heat map is that the drug dosage dictates the extracellular profile. So how nutrients are taken up, that is dictated by the different concentrations of the drug. Whereas the intracellular profile is dictated by the cell type. The cell type determines the intracellular response, which kind of is intuitive as well, but it was nice to see it come out in the, the PLSDA and the PCA analysis. So, okay, all this is good, right? This I have told you a nice story so far, lots of experiments. Now, how do I connect it with my model? So we obviously wanted to make a context-specific model that defines both our sensitive cell type and our resistant cell type. We had probed, so we had a metabolic network, right, that we used from literature. We had gene expression data, limited gene expression data. We had microenvironment information in terms of the way cells were taking up nutrients and giving out nutrients. Right, and then metabolic profiles, uptake rates. Then we had the respiration data. We had a lot of respiration data to understand how cellular reductase activity changed across different substrates, across different cell types. And we knew what was the growth rate and the biomass that we were forming, the growth yield of the cells. We also went on to look at what was the macromolecular composition and created an equation that represented the growth, that artificial equation that I talked about that you can optimize. We did that in-house and looked at the macromolecular composition and created a biomass composition equation. And using this, we try to understand respiration, flux variability, metabolic flux plasticity, and the metabolic consumption release profiles for an integrated analysis of the glioblastoma cell. So I went through this in the previous hour, so I'm not going to go through it, but this is just for a recap, right? You know that this is what I do for a living. And then these are the kind of maps that I create. And I love the Recon 1 model, although the Recon 3D is what we are currently using because it's the most extensively curated and the largest model that is possible. And Swagatika must have talked a lot about it in her lecture last Friday. But I personally like the Recon 1 model for multiple reasons. My human brain can wrap around 3,300 reactions, but not, more than that. So this work, when we did this work, we used this model, but we reconciled it with Recon 2D because that was what was available at that time. And so we had our core metabolic network. We included exome sequencing data as constraints, metabolic uptake as constraints, the growth rates as constraints, and we ended up, analyzing the difference or the differential reprogramming that takes place between U87, the cell line that is sensitive to temozolomide, and NSP, which is resistant to temozolomide. So I'll give you, I think, 1 or 2 examples. 55, how many more minutes do I have? 10 minutes, okay. Yeah, so one of the constraints-based methods that I talked to you about in the previous lecture was flux sampling. So flux sampling essentially provides information on the range of feasible flux solutions that are possible and also what is the probability of their occurrence. So we used flux sampling to span the entire solution space, and we identified what was the difference in rewiring between U87 and NSP. And the top candidates for us were glycolysis, obviously. So U87 uses glycolysis, whereas NSP tends to use TCA cycle more. There were differences in the malate aspartate shunt, which is basically an anaplerotic reaction network, a network cycle or shuttle, that's the right word. Then folate metabolism, oxidative phosphorylation, cholesterol synthesis, and pyrimidine biosynthesis. So these were the 6 pathways that we saw major reprogramming in. It's shown here. Yeah, so this is the difference in the flux distributions across each of these reactions. So the malate aspartate shuttle is responsible for the indirect transport of oxaloacetate into the cytosol, and it also eventually impacts the NADH concentrations. Folate metabolism impacts the methylation and DNA synthesis damage and all. So based on the reprogramming that we saw, the pathways that were reprogrammed, we identified a chemosensitive panel to check, are there differences in the way U87 and NSP respond to other drugs? And so, so we found that there were differences in 14 drugs. We went on to, this was in a high throughput format, and then we went on to look at a lot of these individually as well. And I'll show you the results of some of these when I talk about the next method, after I explain the next method. So how many of you are familiar with the concept of synthetic lethality? All right, so again the 5%.
Workshop before, so I don't know.
Okay. So it should have been more. Yeah, so for those who do not know what synthetic lethality is or do not remember, if there's a normal cell and there are two genes, gene A and gene B associated with it, and if gene A is non-essential, so that means when you delete gene A, the cell will survive. And when you delete gene B, the cell will survive as well. But when you delete both gene A and gene B, then the cell is going to die. Okay, so the cell cannot survive at that point. So these two genes are required together in combination, but individually they are not essential. Now, in the case of cancer, this gene B might not be deleted completely, right? It might just be mutated, and similarly with gene A as well. So a mutation that causes the deleterious effect can be considered as a gene combination that can kill the cancer cell. And for us, this is very useful because, although we talk about, so we want to kill the cancer cell, right? We're really not interested in keeping it alive. And in our case, we want to kill both NSP as well as U87. We want to kill we want to continue to kill the cell type that is sensitive to temozolomide, and we also want to kill NSP, which is not as sensitive to temozolomide. There's another concept that is tied to synthetic lethality, which is called positive epistasis. And in that, if you delete gene A, there's cell death happens. If you delete gene B, it is non-essential, so cell will survive. But if you delete both gene A and gene B, then the cell still survives. So this is sort of gene B, becoming a rescuer of gene B becoming a rescuer of the deletion of gene A, which causes cell death. So this process is called epistasis. So we wanted to use epistasis to understand how drug resistance comes up and use synthetic lethality to understand how we can kill the cell. Because, we were hoping that if we do both together, we should be able to kill all the cells, all the tumor cells. So long story short, we did double gene deletion analysis to understand how many pairs of synthetic lethals exist, how many pairs of PEI exist. We see that U87 has, this is U87, right? So U87 has 16 pairs of PEIs, whereas, why are they two? Yeah, so this is in the presence and absence of the drug. So we did a double gene deletion analysis in the absence of the drug and in the presence of the drug. And we see that in the presence of the drug, there are many more ways that U87 can become resistant because the PE pairs increase. Whereas NSP has potentially evolved ways to become resistant. And that's why the PEI pairs, positive epistatic pairs, in the case of NSP without the drug and with the drug are similar. And we actually typed and looked at are they unique or common, and everything was common between the two. In terms of synthetic lethal pairs, it was interesting to note that the numbers are quite similar when you add the drug and when you do not have the drug. So I'll just take an example of one condition here, one synthetic lethal pair, and that was the SLC5A marker was synthetically lethal with all these genes. And if you look at all these genes, they all belong to the electron transport chain. And SLC5A is essentially a carbon transporter. It's basically a substrate transporter. So you can take in a lot of nutrients using this SLC5A. And as I showed you earlier, there was a differential profile in the mutations that occur for uptake of nutrients, and there was also a differential profile in ETC as well, right? So we do not know exactly, so again, we cannot predict causality here, but we can say that it is correlational. And similarly for NSP as well. So then we went on to pick up, so we've now, so rotenone is a complex 1 inhibitor of the electron transport chain. And so we went on to check what were the IC50 values for this particular drug against U87 and NSP. and found that for NSP, the cell line that was resistant to temozolomide, the IC50 was only 5 nanomolar, whereas for U87 it was 1.8 micromolar. So there was a difference of 3 orders of magnitude between the IC50 values. So this was very heartening for us because we thought we had validated our model. Now we've looked at other electron transport chain inhibitors as well, and we see an interesting pattern in that. Based on the mutational profile. So you can actually now create a map, a matrix map of mutations, electron transport chain complex inhibitors, and what are the IC50 values, and so on and so forth. So, maybe in my next lecture, I will talk about it, not today. Similarly, this is one example of the positive epistasis, and whatever is depicted in green here are potential rescuer genes, and what is depicted in blue is the essential gene. So yeah, these are a lot of genes that I think it will take a day to just explain the pathways that they belong to. So to cut the story short, they are all involved in cholesterol metabolism in one way or the other. So we decided to go ahead and use cholesterol inhibitors. So this is the cholesterol synthesis pathway. Atorvastatin inhibits the HMG coenzyme A reductase, which is in the upper arm of the cholesterol pathway. And berberine is also an inhibitor which inhibits at the lower end of the pathway. And as you can see here, there's a difference in the IC50 values of U87 and NSP. U87 requires only 5 micromolar of atorvastatin, whereas NSP requires 10-fold higher. Similarly, in the case of berberine, you require lesser amounts of lesser amounts of berberine to kill U87 as compared to NSP. So here for berberine, for the lower arm of the pathway, it's 2-fold difference, and for the upper arm, it's a 10-fold difference. So essentially this proves that the hypothesis that was developed using the model and validates our model predictions. So in summary, we've seen that an interplay between metabolism and methylation of glioblastoma genome to shape temozolomide drug response. We've identified metabolic reprogramming in response to temozolomide. And we've identified that it is dose dependent in case of the extracellular impact or the exometabolome profiles, and it is cell type dependent based on the intracellular profiles. We've tried to understand the genetic basis of metabolic adaptation and characterize that using exome sequencing. We've looked at nutrient profiles and understand how they can potentially influence methylation and epigenetic regulation through the temozolomide response. We've looked at an integrative analysis of all this data in the context of a constraints-based human model of metabolism and identified reprogramming using Monte Carlo sampling. And we've also identified using synthetic lethal analysis that NSP is vulnerable to drugs that target oxidative phosphorylation and that using U87 as the model, cholesterol metabolism is implicated in resistance through positive epistasis. So these are my overall conclusions that such integrated systems level approaches are critical to unraveling a lot of tacit connections. It's not enough to just list out catalog, but it's necessary to integrate them in platforms. And this is just one example, one platform modeling strategy that we've used here. So acknowledgement to all my funding sources, DST, DBT, CSIR, the Bill Gates and Melinda Gates Foundation, Rockefeller Foundation, and Hindustan Unilever. Did my students come up? Yeah. So I have to acknowledge Rupa and Avinash for a lot of the work that was done on the metabolic reprogramming. Sarupa is currently a project scientist in Institute of Systems Biology at Seattle, and Avinash is a project scientist at Francis Crick Research Institute at London. And Vijendra and Madhuri are two very talented students who have taken up this work, and they've done the synthetic lethality work that is in progress, is being done by them. And yes, in the end, I'll leave you with a picture of our group, and I would be happy to take any questions.